Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. This is Dr. Satish Devanapalli, working as assistant professor in the Department of Pharmaceutical Science and Technology at Institute of Chemical Technology, Mumbai. So, in previous lecture, we started the topic nanoparticulate as a drug delivery systems. In our previous lecture, we have seen the polymer based nanocarriers, and in that we have discussed on the introduction on the polymer based nanocarriers, and after that we have seen what are the polymeric nanoparticles. In that we have seen the nanospheres and the nano vesicles. So, in that we have seen various preparation methods such as solvent evaporation and the solvent emulsification or solvent diffusion method, and also we have seen the nanoprecipitation method. And after that we have seen the what are the polymeric micelles. We have seen the examples of polymeric micelles, and then we have seen the what are the dendrimers, and we have seen the importance of the dendrimers, and then we have seen the polymerosomes. So, in my today's lecture we are going to discuss on the lipid based nanocarrier systems. So, this is my flow of the presentation. First we will see the introduction on lipid nanocarriers, and in that we are going to see the one lipid based system which is liposomes. What are the liposomes? And we will see what are the advantages also, and further we will see the classification of lipid nanocarriers, and after that we will see what are the types are available in the liposomal formulations, and then we will see the preparation methods for the liposomes. As we know, lipid based nanocarriers can be classified based on their structures and their size. Now, here are the few examples. So, first category is the liposomes, and second is solid lipid nanoparticles, and third one is nanostructured lipid carriers. So, with the introduction on the lipid based nanocarriers, so we will see what are the lipid nanoparticles. Lipid nanoparticles are the nanoparticles, the size range from 10 to 1000 nanometer, which are made up of the lipids that can carry the drugs or genetic materials into the body. So, in previous lecture we have seen the polymeric nanocarriers, and based on the material properties we know that which can be classified in different systems. First is polymeric nanocarrier systems based on the polymer nature, and further we are seeing the lipids. Lipids, yes, you can see the various classification of the lipids. Lipids can be classified in by the based on their properties and their source, fatty acid source and the steroids, and further it goes on. So, these are the lipids we use in the various formulations. As in conventional formulations, when you come into the novel drug delivery systems, which lipids we use for the converting into the formulations. So, definitely which will carry the molecules and which will reach into the target site, and there they will deliver the molecules in the body. And as we seen in case of, we can compare with the polymer nanoparticle, there also we have seen there are the FDA approved polymers. Now here also lipid nanoparticle have gained interest in the field of medicine, which is not limited to the pharmaceutical and not limited to the biotechnology and not limited to the nutraceuticals, which extends to the cosmeceutical applications also. So, what is the main reason? That because of their safety and the non-toxic profile. So, which they have the approved by the FDA for the drugs and the vaccine delivery. So, now we will see the how these can be classified based on their structure, and the composition of lipid nanoparticle vary and depends on the types of lipid and method of synthesis. We will see various structures and compositions of the lipid nanoparticles. So, in this class today we are going to learn on the liposomes. In upcoming classes we are going to learn on the solid lipid nanoparticles and the nanostructural lipid nanoparticles. So, in the liposomes, yes, we know that which are the made up of by the lipids. So, here these lipids form a spherical shaped bilayer vesicles, which are nothing but liposomes, is composed of one or more phospholipid bilayers which closely resemble the structure of cell membranes that are capable of encapsulating active drugs. So, scientists and researchers, they mimic the biological structures. You can also say these formulations are the biomimic carriers, because the cell membranes also contain the lipids. Based on that, scientists discovered these liposomes as a nanocarrier system for delivering of the and encapsulating of the drugs. The bilayer structures are called as liposomes, whereas in previous lecture also we have seen micelles are the monolayer structures. Now, in the structure you can see here in the figure, you will see here the bilayer structure. So, it contains the two layers. Now, yes, these are the individual units, lipids, and when you disperse in any medium by applying some of magnetic stirring or by some heating, so you can convert these lipids into the liposomal vesicles. So, you can see the structure of that lipids, which contains hydrophilic head and the hydrophobic tails. When you disperse in a water, which forms a self-assembling bilayer structures, so which may contain in a water formulations. Now, these are the some of the lipids examples. Here you can see the natural phospholipids and the synthetic phospholipids. So, phosphatidyl-choline, phosphatidyl-ethanolamine, phosphatidyl-serine, and you can see various phosphatidylcholines which comes from the synthetic nature. So, due to their size and the hydrophobic and hydrophilic character of these properties, liposomes are promising systems for the drug delivery. You can encapsulate both hydrophilic, hydrophobic drugs into the one system. Now, here we will see liposome based drug delivery, how this loading will happen, how this lipid or liposomal formulations will show the effect in the human body. Here we learn the mode of action and how this liposomal formulation will help in the prolonging the half life of the molecules. Now, you can see as we learn that liposomes are the spherical shaped bilayer vesicles. Now, here if your drug is hydrophilic or water soluble molecule, you can consider any biological molecule. So, it contains aqueous core which will encapsulate the hydrophilic molecules. If your drug is hydrophobic, which is not soluble in water, which are lipophilic in nature. So, the in between this bilayer, it contains hydrophobic tail, so which can encapsulate your hydrophobic drugs. With this, we will understand that in a one system you can put hydrophilic drugs, water soluble drugs and hydrophobic, lipophilic, water insoluble drugs in a one system. That is a great advantages compared with the other systems. Now, yes, you can also conjugate with various polymers, you can also modify the surface with the monoclonal antibodies and also with the ligands. You can also modify the overall surface charge or surface of the liposomes. With this, you can encapsulate variety of the biological molecules, drugs and protein peptides in a one system. Now, after administration, we will see what is the fate of the liposomal formulation in a human body. Yes, after administration, it undergoes two types of the mechanisms for the cell uptake by using endocytosis and the membrane fusion. So, this is the cell, it will taken up by the endocytosis or by the membrane fusion mechanism. After further it undergo it due to the endosomal lysosomal pH in the cell. So, that will degrade the your lipid vesicle and later your drug molecule will be released and it will show the action on the cells. And now, how these systems will help in increasing the half life of the formulation. As we seen in the previous lectures with the pegylation, polyethylene glycol will help increase the half life of the most of the nanocarriers. Now, you can see this is the polyethylene glycol, this is your liposomes. By conjugating of PEG on the surface of the lipid, which can enhance the half life by inhibiting the opsonization method or bypassing the reticuloendothelial systems. This already we have seen. We can also target by active targeting mechanism by labeling with the carbohydrates and other molecules, antibody with the cleavable linkers and directly with the antibodies. With this, you can also use these liposomes for targeting the as in cancer therapy and other infectious diseases based on the ligand properties. Now, here we know that liposomes contains lipids and as well as cholesterols, because cholesterol has a critical role or crucial role in the formation of the bilayers. Now, you can see this structure. So, here these are the phospholipids. So, it is a bilayer vesicle, right, and to convert as in a bilayer. So, cholesterol has a crucial role in the formation of the self assembling of these lipids. Now, this cholesterol works as a packing agent in between these phospholipids. So, with that, which will increases the integrity of the membrane and also it will enhance the stability of the liposomes. So, in conclusion, we can say that it will regulate the membrane fluidity, it will decreases the permeability of the membrane as well as enhancing its structural stability. And one more greater advantage is that which the cholesterols prevent the leakage of the encapsulated materials through the membrane. So, because of this cholesterol will makes the liposomal vesicles into rigid, so which will control the release, or you can say the leakage of the encapsulated molecules, whether it may be water soluble drugs or water soluble biological molecules from the inside to outside. So, that is one of the great advantage of the cholesterol in the formulation of the liposomes. Now, here we are going to see the classification. So, here first we will discuss on the classification of the liposomes, then in the liposomes we are going to see the types of the liposomes. Now, liposomes classify into two categories, one is unilamellar vesicles, multilamellar vesicles. Based on their layers or based on their vesicles, number of vesicles, which can be classified into two categories. You can see here the first one is the unilamellar vesicles, and then you can see the multilamellar vesicles. So, here in the unilamellar vesicle, this vesicle has a single phospholipid bilayer. You can see single phospholipid bilayer, but which can further classified based on their size, nothing but small unilamellar vesicles, large unilamellar vesicles. We can see here also in the table small unilamellar vesicles and the large unilamellar vesicles. So, based on their lipid layers and the size, so small unilamellar vesicles contain the size from 20 to 100 nanometer, whereas large unilamellar vesicles contain more than 100 nanometer. So, based on that, the unilamellar vesicles are classified into two categories, small unilamellar vesicles, large unilamellar vesicles, and further it classified into multilamellar vesicle, which contains onion type structures. Now, you can see there are the several layers in these vesicles, and you in the right side you can also see the number of layers are increasing. So, here in the multilamellar we have 5 to 20 lipid bilayers, whereas in case of oligolamellar you will see very few type of the layers, considerable bilayers, that is nothing but 5 bilayers. Now, the size is 0.1 to 1 micron, whereas here the size more than 0.5. So, here in oligolamellar you will see very few layers on the structure, whereas multilamellar it looks like a onion like structure and the layers are more than 5, it goes to 20 lipid bilayers. And the last one is multivesicular vesicles, which contains in the one vesicle they have the different vesicles in a one vesicle, which is nothing but multi-compartmental structures. So, on the basis of their size and the layers, it can be classified into different categories. Now, we are going to see the types of liposomes. So, here first is conventional, second is the cationic liposomes, third is peg coupled liposomes, fourth one is the immunoliposomes. So, we know that liposomes consist of the lipids, and because of in their head group contain the phosphate group, so which shows the negative charge on the surface. So, the drawback we know that because of the when you want to go for the high cellular uptake, because cell membranes also contain negative charge. So, the cell membranes also has a negative charge. You will see the repulsion between your particle and the cell membranes, that leads to the decreased cell uptake. So, to avoid this, these conventional carriers must be converted into the cationic to enhance the cell uptake in the body. So, that is why scientists have discovered the cationic lipids. They modified their structure by adding the amino groups into their lipid structure. So, which shows the cationic charge. whereas conventional they have the negative charge, and the half life of the conventional liposome is very less, whereas cation liposomes have a positive charge, their cellular uptake is very high, and the half life of the interaction with the cell membrane or cells is very high. So, further to avoid the phagocytosis or short half life of the liposomes, people have discussed on the pegylated liposomes. We already seen which contains the OH groups on the surface of the polyethylene glycol, which reduces the reticuloendothelial system, which that we will see the improved half life in the pegylated liposomes. And in the fourth class, we are going to see on the immunoliposomes. Wherever you want to target to particular cells, you can conjugate your liposomes with the monoclonal antibodies and other type of the ligands, which can be used for the specific targeting for the anti-cancer and other infectious diseases. So, here we are going to see the methods of preparation. So, we know that in the liposomes, lipids are the major raw materials and components. Now, in the picture you can see, first we need to disperse or dissolve your lipid in the required medium, especially in the different organic solvents. Once you dispersed, and you need to remove the solvent by the drying technique, and after that it will form in a layers, it will form a thin film on the surface of the glass containers, especially here the RBF. So, once drying step is done, after formation of thin film, you need to hydrate with water of your thin films, and then once you are done with hydration, you need to give some agitation or some high pressure homogenization, so that your lipid will start forms the self assembling particles, which are nothing but liposomes. So, further you will get in a required liposomes, you need to further purify, and then you can go for the characterization. Now, which can be divided into the 4 steps. So, after dissolving, you need to do the drying of the lipids, and then disperse in a water, that is hydration method, then purify and go for the further analysis. So, this is a general method for the preparation of the liposomes. Now, we are going to see some of the specific methods. So, here it can be classified into different categories. One is a passive loading and another is active loading. So, here we load the drug after the preparation of the blank liposomes, whereas passive loading, these are in situ, where we load the drug during the preparation method. So, here this can be classified into mechanical disruption method, solvent dispersion methods, and the detergent removal method. So, few methods we are going to see. For example, we are going to see ethanol injection method, and also we are going to see a thin film hydration method, and we are also going to see the sonication method. So, we will focus on few preparation methods. Now, here in thin film hydration method, now you know, with this you will understand. So, what is the general preparation methods of the lipids. So, here in organic solvent, you need to dissolve your lipid first. If a drug is water soluble, you must dissolve in your aqueous solution in a second step of during the hydration. If a drug is water insoluble or lipophilic, lipid soluble drugs, which you can disperse in the stage 1. So, once dissolve your lipid and the drug, and further after dispersion you need to remove the solvent. So, that you can done by the evaporation technique or any other techniques. So, you need to make a thin film after the drying of the solvent, then you can hydrate your thin films with your water or any aqueous systems, and after that you need to do agitation by simple mixing or by vertexing method. So, you will get a large multilamellar vesicles. So, which is not suitable for whenever you want to go for the as an parental application, and if you want to reduce any particle size, you must go with some disruption technique, that may be sonication or extrusion method. Here the layers are very high compared to small unilamellar vesicles and the size is very high. So, you need to do the sonication or disruption method, you need to apply some energy. So, that can be converted into small unilamellar vesicle, especially from 10 to 100 nanometer of particle size. After that you can do the purification. If you want to read more, you can go through this link. So, here the same method by handshaking method. So, here what we do, instead of applying the rota evaporation or by using heating method. So, what we will do, we will disperse our lipid in a solvent and we do the handshaking method, and here also if you want to go further to remove the high amount of water. So, after handshaking you can put under the rota evaporator, that will remove the water, you can apply vacuum and nitrogen to exhale the reaction, and once you get the dry film, disperse with the high water and you will get a liposomal dispersion. This is a handshaking method. And third one, here is the ethanol ether injection method. In both cases ethanol ether are the solvents that we need to dissolve your lipid in the ethanol. So, and then that transfer into the aqueous phase with the injection. So, in ethanol ether injection method, so you need to dissolve your lipids and if required some of the surfactant and dissolve and then inject into the aqueous solution, there you will get a vesicles. But if you want to reduce further, you can go for the sonication or high pressure homogenation, high shear homogenation method or ultrasonication method. So, now this is a mechanical dispersion method. So, here what we do, so same method, you need to dissolve your drug and lipid in a organic solvent and remove the water, you will get a thin film and add your aqueous phase, but after hydration you will get a large particles and you need to do any of these techniques, sonication, extrusion or homogenation method. With that you will get a small unilamellar vesicles. So, with this we will understand, based on the nature of your lipid and the nature of your drug and the type of carrier, and you can select required method. So, with that you can get in right liposomal carriers. And this slide speaks that about the commercial liposomal formulations which are available in the market. Now, here you see these are the various product names. So, here we can read representative list of the liposomal based drug products. Now, here you can see the products and you can see the drugs and the company name and what are the therapeutic use of that molecules. So, these are the various products and these are the molecules. Most of them are the antifungals and the anti-cancer drugs also available for the treatment of the various diseases, and these are the companies. And now, here we will see what are their therapeutic application. You can see the leukemia and these are the various leishmaniasis and infectious diseases, and also you will see lot of cancer therapy. So, with this you can encapsulate various types of the molecules in a liposomal formulation. So, with this we will stop the lecture here. Thank you so much.